Bacteria looks like this and has a bacterial chromosome that has a bunch of DNAs in it, and plasmid, which has just little DNA pieces, and this encircling is the cell envelope, and these small dots are ribosome. What they do is they convert RNA into proteins. On the exterior, we have these little appendages. Flagella is what allows them to like move around, and pilus is fimbriae, and they're a little different, and I'll explain further. The flagella kind of looks like this. There's a filament, and if this is the cell body of the bacteria, we have this basal body, and then hook, and then filament. It depends on the location of or how the flagella is attached to the bacteria. We have classified them into differently. So if it's just like one flagella in one side of the body, it is monotrichous, and lophotrichous if there's multiple on one side. If it's on like both sides, it could be amphitrichous. If it's like everywhere, like E. coli, it would be peritrichous. And for fimbriae and pilus, fimbriae, we have a whole bunch of them, and they're shorter, right? So they have shorter in length and smaller in diameter, but they have more of them, and they are made in bacterial chromosome, and they serve a function of they use those fimbriae to attach to different cell surface, and they're most common in gram-positive and negative, so in both type of bacteria. Whereas pilus, it is a long little tube. So it's longer, but it has less number of them, but they're bigger in diameter, and they're made in plasmid, and they are used as a conjugation pipe. In other words, they use this pipe to convey a plasmid or different genetic material into different bacteria, and most common in gram-negative bacteria. Bacteria can form endospores when the environment is very harsh to increase their survivability. They give more resistance to heat, UV, lack of nutrients, etc. So they, from regular bacteria they become endospores, which has a bacteria chromosome inside, and it's circled by the double layer wall, which are filled with peptidoglycan, and then keratin layer on the outside. And to talk more about the cell envelope. A cell envelope is, we have this either capsule or a slime layer, and it's something that's kind of covering the bacteria. So polysaccharide layer of the cell envelope is if capsule is like organized, and its function is to like make them harder for white blood cells to eat the bacteria. And on the other hand, we have this slime layer of cell envelope, and its polysaccharide layer is loose, and it allows them to adhere to like foreign object better and helps with biofilm formation. And these are the gram-positive and gram-negative bacteria's cell wall structures. So we have on the outside, there's some cell envelope, whether it be capsule or a slime layer. On the inside, this is the inner membrane, and the gram-negative is the only one that has the outer membrane. And both of them have peptidoglycan layer, which are composed with NAG and AAM. Gram-positive have this like LTA, which causes white blood cells to release like cytokine and cause more inflammation. And we also have this, so LTA goes all the way inside to the inner membrane, whereas the teichoic acid does not go all the way into the inner membrane, and it also causes a white blood cell to release more like cytokines and cause inflammation.